Okay, so we have talked about enzymes, and now we're going to immediately go ahead and show a little bit, in very basic terms, how enzymes are very important in chemical reactions. And one of the most important chemical reactions taking place in our cells, which maintains the life of our cells, is the production of energy in the cell. Now, the production of energy in cells is the production of ATP, or adenosine triphosphate. That is the battery power, if you like, that exists in the cells. These molecules carry high energy in the final bond between the second and third phosphate groups. And when that's broken, that energy is released from the bond right at the point where it's required by any given reaction, and then that reaction takes place, or that protein changes shape and then allows something in or out of the cell, etc., etc. Without ATP in the cell, we would not live very long, or that cell would not live very long. So it is an absolutely crucial part of any living system. Cellular respiration requires two main ingredients for it to work en masse. The first is oxygen, and in the second of these two concept lectures, we're going to talk about the role of oxygen and why it's so important. The other one is an energy source. Now, in cell respiration, we can use carbohydrates, fats, and proteins to make ATP, but the system is better designed to use glucose, which is a simple or monomeric carbohydrate, than it is to use fatty acid chains or proteins per se, although it can use all. When we look at it chemically, we always use glucose at the early stages, because that's the main player in the game and shows us the entire process of cellular respiration, which occurs in 3 stages. Now, in this particular short talk, we're just going to look at the first stage, which is called glycolysis. And in this stage, it occurs inside the cell, but outside of the mitochondrion. In fact, in cells like red blood cells, which do not contain mitochondria, when they reach maturity, then this is the only one of the 3 phases which do occur. Now, in the 3 phases, they all produce ATP, but the first 2 phases, glycolysis, and the second phase, Krebs cycle, produces a very low amount of ATP, just 2 ATP per glucose molecule, whereas the final stage produces anywhere between 25 and 38 ATP molecules. So you can see the advantage of having mitochondria and having the oxygen to allow the whole process to take place, because you're getting 16 times the amount, maybe even more, than you would do without using the final stage. However, we're going to start with the glycolysis and look at what that means. It occurs in the cytoplasm of the cell, so it's occurring at a pH of 7, because that's the pH of the cytoplasm of living cells. So behind me, I've written up some of the chemistry. You don't need to look at this and say, oh my god, I have to learn this, because you don't. But I'm showing you from the chemical level, just to, it's easier for me actually, to look at it and remember what I want to tell you. So, this is cell respiration. We breathe, we call it the respiratory system. We're breathing in oxygen and breathing out carbon dioxide. The reason why we do this is to drive this reaction. We take in oxygen to produce the energy. We release carbon dioxide because it is a byproduct of producing energy. And as we'll show later in the course, carbon dioxide and water mix to form carbonic acid. That's an acid that changes the pH. If it builds up, it stops reactions from taking place. So we breathe the carbon dioxide back out to stop our blood and our cells and our interstitial fluids from becoming acidic. Alright, so the term glycolysis is 2 parts. Glyco, relating to glucose and carbohydrates, and lysis, meaning to break. So what are we going to do? Well, we're essentially going to break the glucose, and in very simple terms, the glucose is broken right in the middle. So you've got 6 carbons forming the backbone, and once again, I'm going to use my very expensive prop. So here's 6 carbons, 3 on the blue, 3 on the purple, and we're going to split it into 2. And these become pyruvic acids, 3 carbon molecules. So good so far. So here's our glucose going into the system. Here's our pyruvic acid after the split. So there's 2 of them, remember. There's one, I haven't drawn the other one on the diagram, but we have 2 of them. To get from here to here, there are 4 enzymes required and 3 intermediary stages before you reach the 3 carbon product. We're doing physiology, we're not doing biology or biochemistry, so we don't need to learn every one of the chemicals and every one of the enzymes and how it changes, going from glucose to glucose 6-phosphate, etc, etc, etc. So we'll just accept that this molecule here, after going through a series of reactions, becomes pyruvic acid, which is a 3 carbon, because it's, remember, it's been split into 2, so now it's half of what it was. 3 carbons, there's 1, there's 2, there's 3. Now, pyruvic acid has a double bonded oxygen on the second carbon, and it has acid, an acid group, COOH. Whenever you see COOH, that's an acid group, because that hydrogen can leave and become a positive ion, that can leave the oxygen as a negative. So we have 3 pyruvic acids, 3 carbon pyruvic acids, we have 2 of them. Now, there are more fates than these 2, but in the human cells, these are the 2 that we care about. If there is oxygen inside the cytoplasm of this cell and there are mitochondria present, which I haven't named and we'll just do so now, if oxygen or sufficient oxygen is present in the cell and there's a mitochondria, the pyruvic acid will go through another change, and it will go from a 3 carbon to a 2 carbon molecule. So we're going to lose another carbon. So a coenzyme A, this is a specialist molecule, comes in, reacts with the pyruvic acid, knocks off a carbon dioxide molecule, is lost, so that's CO2, there's the CO2 right there, and the hydrogen from that acid group ends up being picked up by an NAD plus molecule. This is a reduced form, so oxidized form, and then it's reduced by having that hydrogen attached. So this whole section here of the pyruvic acid, the C and 2 O's become carbon dioxide, and the hydrogen is picked up by this little taxi, which we're going to talk about a lot more in the next one. These are very important because these carry these hydrogens, and it's the hydrogens, and remember all a hydrogen is one proton surrounded by 1 electron. Remember, that's important. So they are picked up by these little taxis, little molecules that can pick up the hydrogens and carry them away. Now if there's enough oxygen present, this NADH molecule, along with, the acetyl coenzyme A, which is what's formed when you take that off, when you break that away there, you add the coenzyme A in its place. So now you've got acetyl, because that's what this group is here, is an acetyl group, coenzyme A, and that ends up going into the inner part of the mitochondria, where the second 2 phases of cell respiration take place, and we're going to talk about them in the second of these 2 lectures. So we started with glucose, we went through a bunch of intermediaries, we ended up with pyruvic acid, which is a 3 carbon from a 6 carbon start, 3 carbon molecule. The 3rd carbon is broken off, carbon dioxide is released, and hydrogen is picked up by the NAD molecule and carried away. So the coenzyme A is attached, and so we end up with acetyl coenzyme A entering into the mitochondrion for the next stage. if there is not enough oxygen present, or there's no mitochondria, which is the case in red blood cells, we can't actually do this because of this little taxi down here. Because at this end stage of glycolysis, what we haven't looked at here is ATP is produced. 2 ATP molecules, and that's net production, are produced earlier in the glycolysis stage, and that's very much dependent on the loading of this hydrogen here. Now if these all get loaded, but they can't go away and do their job because there's not enough oxygen here to allow them the next stage to take place, then the whole system stops. And if the whole system stops, you've got no glycolysis, you've got no ATP production, your cell is going to die. So if there's not enough oxygen, you can't continue on in this way because this will not lead to any energy production without the mitochondria doing the second two stages. So there's an alternative pathway for it. Now there's actually more than one. In plants, this is how we make alcohol. It's called the fermentation pathway. We use glycolysis in yeast to actually produce C2H5OH and that's the alcohol. But in our cells, probably just as well, we don't produce alcohol because, you know, as soon as we went aerobic respiration, we'd start all getting drunk, which would be interesting but not very productive. Instead, we produce a different molecule. Instead of acetyl coenzyme A, we produce lactic acid. So if we're looking back at this pyruvic acid molecule, instead of just lopping off the whole of the acid part here, the COOH, we maintain the COO part of it, but we lose the hydrogen ion. So the hydrogen ion comes off. But we split the double bond here for the oxygen and we add a hydrogen in there, and because we've got another bond available with another hydrogen there. So this is lactic acid. Now the importance of doing this process, this step from here to here, allows a hydrogen to come off the NADH, because if you think about it, if you look at this molecule here and this molecule here, this has a hydrogen here, and this has a hydrogen there. That's the only differences. So you can rearrange this molecule by taking your hydrogen and putting it on that oxygen, but you still need a hydrogen for the other one. And that comes from the NADH. And so you're offloading it and you're turning the NADH back to NAD+, which can go and pick up another hydrogen from earlier in the reactions, and that allows it to make ATP at that stage. So by producing lactic acid at the end of this process, if there's no oxygen present or there's no mitochondria to take on the next two stages, you prevent the NAD+ from running out. And that allows the earlier system to keep producing ATP. Now I don't expect you to understand all that in any great detail, simply because we haven't taken the time to go through the actual biochemistry of it. But for us, there are two pathways. Glycolysis starts with glucose, goes through a bunch of intermediaries and produces pyruvic acid. One, if there's enough oxygen and there's mitochondrion present, that will continue on, we'll lose the acid group, add a coenzyme A, and that will head into the Krebs cycle. Two, if there is not enough oxygen, then this cannot take place because it would shut down all ATP production. So instead, we have a rearrangement of the pyruvic acid molecule to lactic acid, which requires one more hydrogen, which we peel off the NADH, which is made earlier in the system, and recreate an NAD+. And so that allows that to cycle back into the earlier produce to keep producing ATP. However, we now have a new problem. We're producing lactic acid. So keep in mind that aerobic respiration is with oxygen and anaerobic respiration is without oxygen. So when we're respiring anaerobically, we're using too much energy in the cells before the oxygen can get there and make new energy. So that's when we end up with pain and cramps in our muscles, etc., because our body's saying, hey, hang on, you're going too fast here, you need to slow down, we're running out of energy. And the lactic acid is what's causing the burn, because it's an acid, it's changing the pH from 7 to lower than 7. So remember, the pH here equals 7, and it's being lowered due to lactic acid. So then the cell takes that and dumps it outside the cell into the interstitial fluids. And that's picked up and goes into the blood, and then starts dropping the pH of the blood, sending the blood into acidosis. We're building up hydrogen ions, and the one way we can get rid of that is blowing off carbon dioxide. So we start breathing faster, which is why we start breathing faster when we're exercising. It's not just because we need more oxygen, but we have to stop our blood and our cells from becoming acidic. Okay, well, I've probably confused you a little bit here. If you have questions about this, I've tried to go through the basics of it. In the second lecture, I'm going to talk about the importance of these NADH molecules, because they're the little taxis that carry the hydrogens around to actually get ATP made. And we're going to look at how it's done inside the mitochondrion. So for this one, you just need to know that glucose becomes pyruvic acid, can go on to become acetyl coenzyme A and enter the mitochondrion if there's oxygen, and if not, it produces lactic acid to prevent the NAD plus from running out, and earlier ATP production from ceasing altogether. That's it. Thank you very much.